Hello students, today we'll be dealing with oxidative phosphorylation part 1, which is chemiosmotic hypothesis. Let's look at the introduction first. The hypothesis that a proton motive force across the inner mitochondrial membrane is the immediate source of energy for ATP synthesis was proposed in 1961 by Peter Mitchell. Mitchell's chemiosmotic theory postulates that the energy from oxidation of components in the respiratory chain is coupled to the translocation of hydrogen ions or protons from the inside to the outside of the inner mitochondrial membrane. The electrochemical potential difference resulting from the asymmetric distribution of the hydrogen ions is used to drive the mechanism responsible for the formation of ATP. Almost all researchers studying oxidative phosphorylation and photosynthesis initially rejected his chemiosmotic hypothesis. They favored a mechanism similar to the then well-elucidated substrate-level phosphorylation in glycolysis, in which chemical transformation of a substrate molecule is directly coupled to ATP synthesis. However, in spite of their intense efforts, compelling evidence for such a substrate-level phosphorylation-mediated mechanism was never observed. Definitive evidence supporting Mitchell's hypothesis depended on developing techniques to purify and reconstitute organelle membranes and membrane proteins. The experiment with vesicles made from chloroplast thylakoid membranes containing ATP synthase was one of the several demonstrating that this protein is an ATP generating enzyme and that ATP generation is dependent on proton movement down an electrochemical gradient. It turns out that the protons actually move through the ATP synthase as they cross the membrane. Now let's look at the experimental findings that support the chemiosmotic theory. Number 1. Addition of protons to the external medium of intact mitochondria leads to the generation of ATP. Secondly, oxidative phosphorylation does not occur in soluble systems where there is no possibility of a vectorial ATP synthase. A closed membrane must be present in order to achieve oxidative phosphorylation. And thirdly, the respiratory chain contains components organized in a sided manner, that is transverse asymmetry, as required by the chemiosmotic theory. Proton motive force and chemiosmotic coupling. The cytosolic phase of the bacterial plasma membrane, the matrix of the inner mitochondrial membrane, and the stromal phase of the thylakoid membrane are all equivalent during electron transport. In the electron transport chain, the protons are always pumped from the cytoplasmic phase to the exoplasmic phase, creating a proton concentration gradient and an electric potential across the membrane. So the main result of the electron transport chain is a generation of the proton motive force, which is the sum of the transmembrane proton concentration gradient and electric potential or voltage gradient across the mitochondrial inner membrane. In chemiosmotic coupling, this proton motive force generated by proton pumping across a membrane is used to power energy-requiring processes like ATP synthesis or flagellar movement in bacteria. The mechanism and membrane orientation of the processes are similar in bacteria, mitochondria, and chloroplasts. Now let's look at the proton gradient-driven ATP synthesis. The mechanism of ATP synthesis is similar among bacteria, mitochondria, and chloroplasts. Although bacteria lack internal membranes, aerobic bacteria nevertheless carry out oxidative phosphorylation by the same processes that occur in eukaryotic mitochondria and chloroplasts. Enzymes that catalyze the reactions of both the glycolytic pathway and the citric acid cycle are present in the cytosol of bacteria. enzymes that oxidize NADH to NAD+, and transfer the electrons to the ultimate electron acceptor, which is oxygen, reside in the bacterial plasma membrane. The movement of electrons through these membrane carriers is coupled to the pumping of protons out of the cell. The movement of protons back into the cell down their concentration gradient through ATP synthase drives the synthesis of ATP. The bacterial ATP synthase is identical in structure and function to the mitochondria and chloroplast ATP synthesis, but is simpler to purify and study. In addition to ATP synthesis, the proton motive force across the bacterial plasma membrane is used to power other processes, including uptake of nutrients such as sugars and the rotation of bacterial flagella. Chemiosmotic coupling thus illustrates an important principle that the membrane potential, the concentration gradients of protons and other ions across a membrane, and the phosphoanhydride bonds in ATP are equivalent and interconvertible forms of chemical potential energy. Here in this figure, we see that energy from sunlight or the oxidation of foodstuffs is first used to create an electrochemical proton gradient across a membrane. This gradient serves as a versatile energy store and is used to drive a variety of reactions in mitochondria, chloroplasts, and bacteria. Now we shall look at electron transport and ATP synthase. The ATP synthase is a reversible coupling device that can convert the energy of the electrochemical proton gradient into chemical bond energy or vice versa. That is, it can either synthesize ATP by harnessing the proton motive force or pump protons against their electrochemical gradient by hydrolyzing ATP. Chemiosmotic coupling is now recognized as a general mechanism of ATP generation, operating not only in mitochondria but also in chloroplasts and in bacteria, where ATP is generated via a proton gradient across a plasma membrane. Electron transport through complexes 1, 3, and 4 of the electron transport chain is coupled to the transport of protons out of the interior of the mitochondrion. Thus, the energy-yielding reactions of electron transport are coupled to the transfer of protons from the matrix to the intermembrane space, which establishes a proton gradient across the inner membrane. Complexes 1 and 4 appear to act as proton pumps that transfer protons across the membrane as a result of conformational changes induced by electron transport. In complex 3, protons are carried across the membrane by coenzyme Q, which accepts protons from the matrix at complexes 1 or 2 and releases them into the intermembrane space at complex 3. Complexes 1 and 3 each transfer 4 protons across the membrane per pair of electrons that is transferred. In complex 4, 2 protons per pair of electrons are pumped across the membrane, and another 2 protons per pair of electrons are combined with oxygen to form water within the matrix. Thus, the equivalent of 4 protons per pair of electrons are transported out of the mitochondrial matrix at each of these 3 complexes. This transfer of protons from the matrix to the intermembrane space plays a critical role of converting the energy derived from the oxidation-reduction reaction of electron transport to the potential energy stored in a proton gradient. Because the phospholipid bilayer is impermeable to ions, protons are able to cross the membranes only through a protein channel. This restriction allows the energy in the electrochemical gradient to be harnessed and converted to ATP as a result of the action of the 5th complex involved in oxidative phosphorylation, which is complex 5 or ATP synthase. This enzyme creates a hydrophilic pathway across the inner mitochondrial membrane that allows protons to flow down the electrochemical gradient. So as these ions thread their way through the ATP synthase, They are used to drive the energetically unfavorable reaction between ADP and inorganic phosphate that makes ATP. ATP synthase is organized into two structurally distinct components, F0 and F1, which are linked by a slender stalk. The F0 portion spans the inner membrane and provides a channel through which the protons are able to flow back from the intermembrane space to the matrix. The energetically favorable return of protons to the matrix is coupled to ATP synthesis by the F1 subunit, which catalyzes the synthesis of ATP from ADP and phosphate ions. The flow of protons through F0 drives the rotation of F1, which acts as a rotary motor to drive ATP synthesis. Thus, the mechanical energy is converted into chemical bond energy through the repeated changes in protein conformation that the rotating stalk creates. In this way, the ATP synthase is able to produce more than 100 molecules of ATP per second. Now let's look at the proton gradient-driven coupled transport. The synthesis of ATP is not the only process driven by the electrochemical proton gradient. In mitochondria, many charged small molecules such as pyruvate, ADP and inorganic phosphate are pumped into the matrix from the cytosol, whereas others such as ATP must be moved in the opposite direction. Carrier proteins that bind these molecules can couple their transport to the energetically favorable flow of protons into the mitochondrial matrix. Thus, for example, pyruvate and inorganic phosphate are co-transported inwards with protons as the protons move into the matrix. In contrast, ADP is co-transported with ATP in the opposite directions by a single carrier protein, which is the ATP-ADP antiporter. Since an ATP molecule has one more negative charge than ADP, each nucleotide exchange results in a total of one negative charge being moved out of the mitochondrion. This ATP-ADP co-transport is thereby driven by the voltage difference across the membrane. Phosphate re-enters the mitochondrial matrix with the protons by an electroneutral symport mechanism. In the eukaryotic cells, the proton gradient is thus used to drive both the formation of ATP and the transport of certain metabolites across the inner mitochondrial membrane. In bacteria, the proton gradient across the bacterial plasma membrane is harnessed by both types of functions. And in the plasma membrane of motile bacteria, the gradient also drives the rapid rotation of the bacterial flagellum, which propels the bacterium along. The chemiosmotic hypothesis consisted of 4 independent postulates. In terms of mitochondrial function, they were as follows. Number 1, the mitochondrial respiratory chain in the inner membrane is proton translocating. That is, it pumps protons out of the matrix space when electrons are transported along the chain. Second, the mitochondrial ATP synthase also translocates protons across the inner membrane. Being reversible, it can use the energy of ATP hydrolysis to pump protons across the membrane, but if a large enough electrochemical proton gradient is present, protons flow in the reverse direction through the complex and drive ATP synthesis. Number 3. The mitochondrial inner membrane is equipped with a set of carrier proteins that mediate the entry and exit of essential metabolites and selected inorganic ions. And number 4, the mitochondrial inner membrane is otherwise impermeable to hydrogen ions, hydroxyl ions, and generally to anions and cations. Several key observations are explained by this mechanism. Firstly, intact inner mitochondrial membrane is required for oxidative phosphorylation. Secondly, the inner mitochondrial membrane is impermeable to ions such as hydrogen ions, hydroxyl ions, potassium ions, and chloride ions, since free diffusion of these type of ions would discharge an electrochemical gradient. Thirdly, the transport of Electrons result in the transport of protons out of the intact mitochondria from the matrix to the intermembrane surface, thereby creating a measurable electrochemical gradient across the inner mitochondrial membrane. Number 4. Compounds such as dinitrophenol, DNP, and FCCP that increase the permeability of the inner mitochondrial membrane to protons and thereby dissipate the electrochemical gradient allow electron transport to continue to inhibit ATP synthesis. That is, they uncouple electron transport from oxidative phosphorylation. Conversely, increasing the acidity outside the inner mitochondrial membrane stimulates ATP synthesis. Now we shall conclude this lecture with the following points. Peter Mitchell proposed the chemiosmotic hypothesis that a proton motive force across the inner mitochondrial membrane is the immediate source of energy for ATP synthesis. The proton motive force is the sum of the transmembrane proton concentration gradient and the electric potential or voltage gradient across the mitochondrial inner membrane generated owing to the pumping of protons across the membrane during electron transport. In chemiosmotic coupling, the proton motive force generated by proton pumping across the membrane is used to power energy requiring processes like ATP synthesis, co-transport of certain metabolites, or flagellar movement in bacteria. Bacteria, mitochondria, and chloroplasts all use the same chemiosmotic mechanism and a similar ATP synthase to generate ATP. The ATP synthase is a reversible molecular coupling device that can convert the energy of the electrochemical proton gradient into chemical bond energy or vice versa. That is, it can either synthesize ATP by harnessing the proton motive force or pump protons against their electrochemical gradient by hydrolyzing ATP. It does so by creating a hydrophilic pathway across the inner mitochondrial membrane that allows protons to flow down their electrochemical gradient. As these ions pass through the ATP synthase, they are used to drive the energetically unfavorable reaction between ADP and inorganic phosphate that makes ATP. And lastly, several key observations in the mitochondrial membrane function are explained by this hypothesis.